Bismillahirrahmanirrahim. Assalamu alaikum everybody. I hope that you are in good health and prosperity. In today's brief communication, we are going to talk about the mechanisms involved in the smooth muscle contraction and relaxation induced by different autonomic drugs. Just we imagine this is the autonomic receptors, which might be alpha-1 adrenergic receptors, or they might be muscarinic cholinergic receptors type 1 or 3. These receptors, they are linked to GQ11 protein. Once the agonist, whether it is alpha-1 agonist or muscarinic type 3 receptor, M3 receptor agonist binds to the receptor, they stimulate G protein, which in its turn will activate a membrane enzyme known as phospholipase C. Phospholipase C will act on a membrane phospholipid, which is phosphatidyl inositol diphosphate, and convert it into 2-second messenger, diacylglycerol or DAG, diacylglycerol, or inositol triphosphate, DAG will act on cytoplasmic protein kinases known as protein kinase C, while IB3 will leak into the cytoplasm and it will bind to specific receptors known as IB3 receptors on the sacroplasmic reticulum. These IB3 receptors, they are linked to calcium channel. Once IB3 will bind to this receptor, calcium channel will be open and calcium will rush out from the sarcoplasmic reticulum into the cytoplasm. Once calcium is free in the cytoplasm, it will interact with a calmodulin, which is present in the plasma, and it will form a complex, calcium calmodulin complex. This calcium calmodulin complex will activate another cytoplasmic enzymes. For example, it will activate, this complex will activate myosin light chain kinase. Myosin light chain kinase. This enzyme is present floating in the cytoplasm of the smooth muscle and it is inactive. But once it binds with calcium calmodulin complex, it becomes active. In this case, it will be high or rich in energy and it will phosphorylate the head of the myosin 2 to phosphorylate myosin 2 head. This will lead to giving energy to the myosin to slide over actin, cross-bridging myosin and actin, and this will result at the end in muscle contraction. Okay, so this is usually what happens when alpha-1 receptors are stimulated or M3 receptors are stimulated on the smooth muscle. So the end result is contraction. Why? Because if we start from the beginning again, just to remind ourselves, stimulation of these receptors will activate phospholipase C, which will convert membrane phospholipid into 2-second messenger, inositol triphosphate or IB3, and diacylglycerol. IB3 will leak into the cytoplasm where it goes to the sarcoplasmic reticulum, stimulating certain IB3 receptors, opening calcium channel. Calcium will leak out, bind it with calmodulin, producing a complex that will activate myosin light chain kinase. Once myosin light chain kinase is activated, it will lead to phosphorylation of the myosin 2 head, and this phosphorylation will result at the end in muscle contraction. Okay, so now this is muscle contraction due to stimulation of certain adrenergic or cholinergic receptors. Of course, this contraction induced by cholinergic receptors is true for all smooth muscles except those of blood vessels. So M3 receptor stimulation of M3 receptor in smooth muscle of the blood vessel will produce relaxation rather than contraction, as we will see in a few minutes. Okay, so now all smooth muscles, like for example alpha 1 receptor on smooth muscle blood vessel will produce vasoconstriction. M3 receptors on the smooth muscles of bronchioles or GIT or the uterus reproduce contraction of smooth muscle of the uterus. M3 receptors on the smooth muscle blood vessel will lead to the relaxation of the smooth muscle, and we will explain that in a minute. Okay, now so what will happen, how muscle is relaxed? So we talked about the contraction. So how muscle is relaxed? Smooth muscle is relaxed by the effect of myosin light chain phosphatase. Myosin light chain phosphatase. There is a missing here of chain. Myosin light chain phosphatase. This enzyme will cause dephosphorylation of myosin 2. Dephosphorylation means removal of the phosphate group, i.e. it will become inactive. Once it will become inactive, that means muscle will relax because there is no source of energy. Okay, so myosin light chain phosphatase, what it will do, it leads to dephosphorylation of the myosin 2 head. Dephosphorylation means removal of the phosphate group, i.e. myosin 2 will become inert. It cannot slide over actin. In this case, it will be relaxed. Okay, so this is the enzyme responsible for the relaxation of the smooth muscle. This enzyme is stimulated by different neurotransmitters. Now, because we are talking about the autonomic, we'll take two examples. The first example is the beta 2 adrenergic receptors. Beta 2 adrenergic receptors. Beta 2, as we know, as we have said before several times, they are inhibitory everywhere. So in this case, beta 2 stimulation, how they produce muscle relaxation. Beta 2 like beta 2 receptors which are present in the blood vessel, beta 2 receptors which are present on the smooth muscles of the uterus, smooth muscle of bronchioles, or are relaxed. Why they are relaxed? Because once beta 2 receptors are stimulated by specific agonists, they will lead to activation of GS protein. GS protein. This is a different type of protein, a G protein. We have said they are of different types. This is the HGS stimulatory, which is linked to adenylyl cyclase, linked to membrane enzyme known as adenylyl cyclase. Adenylyl cyclase. Adenylyl cyclase will act on adenosine triphosphate, ATP, adenosine triphosphate, converting it to the second messenger, cyclic AMP. Cyclic AMP. This is the second messenger. Cyclic AMP now in the cytoplasm, it will stimulate another cytoplasmic receptor known as protein kinase A. Protein kinase A, it will become rich in energy and in its turn will activate myosin light chain phosphatase. It will activate myosin light chain phosphatase. Okay, so now what will happen? Once the agonist, beta 2 agonist, bind to the receptor, it will stimulate the GS protein. These GS protein, they are linked to adenylyl cyclase. Activation of adenylyl cyclase, a membrane enzyme, will convert ATP to cyclic AMP. Cyclic AMP, this is the second messenger, which will activate in its turn protein kinase A, and protein kinase A will activate myosin light chain phosphatase. Myosin light chain phosphatase, as we have said, it will dephosphorylate the myosin 2 head, and this will result in relaxation of the smooth muscle. This is one example of a membrane receptors. We have said that M3 receptors they don't, in blood vessels, M3 receptor stimulation blood vessels, they don't result in contraction but in the relaxation. Why this happens? Because when we stimulate the M3 receptors on the smooth muscles of the blood vessels, they will lead to the release of nitric oxide from the endothelial cell. This nitric oxide in the cytoplasm of the cell, it will lead to stimulation of guanylyl cyclase. This guanylyl cyclase will convert guanosine triphosphate into guanosine monophosphate. Okay, guanosine monophosphate, like a cyclic adenosine monophosphate, guanosine monophosphate, it will activate protein kinase A, and protein kinase A will activate myosin light chain phosphatase, causing relaxation of the muscle. So that's why stimulation of M3 receptors on the smooth muscle of blood vessel will result in relaxation of the smooth muscle blood vessel. So M3 stimulation everywhere in the body produce contraction of the smooth muscle except those of blood vessel. Why? Because in the blood vessel, stimulation of M3 receptor will release nitric oxide from the endothelial cover of the blood vessel. Nitric oxide will stimulate guanylyl cyclase, which will convert GTP to cyclic GMP, and cyclic GMP will stimulate cytoplasmic protein kinases, resulting in deactivation of the myosin 2, and deactivation of the myosin 2 leads to relaxation of the smooth muscle. Okay, so these are the mechanisms involved in the contraction and the relaxation of the smooth muscles induced by autonomic drugs. The details of the action produced by these mechanisms are given in my presentation, which you can find in this link. I would like to thank you very much for your attention. Please don't forget to press like. If you have not subscribed to my channel, please subscribe now. I wish you a good health and prosperity. Wassalamu alaikum wa rahmatullah wa barakatuh.